Hello and welcome back to another episode of FTS tutorials. We're still on under food tests. Here we'll be talking about test for protein. Don't forget to like, subscribe. If you're new, hit the notification bell so that you always get notification when we post a new video. And the topic in your mind, drop it in the comment box. We'll treat that in the next video or so. In the millions test is the first test for protein. Protein gives a red precipitate when warmed with millions reagents. Millions reagent is prepared by dissolving mercuric nitrate in nitric acid and then adding water to dilute it. So we know now that protein gives a red precipitate with millions reagents. The condition here is simple. It must be warmed. If you mix it without warming, it will not give you a red precipitate. So in your exams or personal experiment, you should know that the mixture needs to be warmed to get the desired result. So if a food sample is warmed with millions reagent, a red precipitate is formed. That food sample is most likely or is protein, because only protein will give that positive result with millions reagents. And the second one here is the burette test. Protein gives a purple or violet coloration when treated with burette reagent. So the burette reagent is a solution of sodium hydroxide or potassium hydroxide and copper, 2 solutions. Here no heating is required. So the burette reagent can either be a mixture of a solution of sodium hydroxide and copper, 2 solutions, or potassium hydroxide and copper, 2 solution, whichever is available. Both hydroxide mixed with copper, 2 solutions will give you the desired protein of violet results with purple or violet result with protein. You don't need heating here. Remember in the millions test just now, when heating was required, we need to warm it to get the desired result. But in the burette test, no heating is required. Once it's mixed with the food sample and the reagent, you get your purple or violet coloration. Then the other one here is the xanthoproteic test. The reagent in this test is a combination of nitric acid, HNO3, and ammonium hydroxide or sodium hydroxide. On reaction with nitric acid, a white precipitate may be formed due to denaturation of protein. So ammonium hydroxide or sodium hydroxide can be used, but nitric acid is compulsory in this test. If it is done, a white precipitate will always be formed due to denaturation of protein. Denaturation here simply means the breaking of the weak linkages or bonds. The bonds may be hydrogen bond within the protein molecule. The natural protein, the bonds that make it, when you break it down, that process is known as denaturation of protein. When this is done, a white precipitate is formed at first. That's when it reacts with nitric acid. Then a yellow precipitate is formed on heating. On reacting with excess sodium hydroxide or ammonium hydroxide, it turns orange on cooling. So if you denaturalize it with nitric acid, which is HNO3, it gives you a white precipitate. Then on reacting with excess sodium hydroxide or ammonium hydroxide, a yellow precipitate is formed on heating and it turns orange on cooling. So here we notice 3 different color change. The first one is when it reacts with nitric acid, you will get a white precipitate. Then on heating with sodium hydroxide or ammonium hydroxide, a yellow precipitate is formed on heating, and when it cools, it turns orange on cooling. So you notice the 3 color changes. These still are the main test for protein. Don't forget to like, subscribe, as I've said, and drop the topic you want us to do next. Till I come your way again. Bye. Bye.